So tomorrow we will go over the lab exam. I'll have some pictures of the results and we'll go through the answers and things like that. Again, I'll be collecting the exams, and so you'll have a sheet that has your score on it, but then I'll be collecting the lab exam. You'll have the remaining part of the period tomorrow to work on anything you need to work on. And then next week's labs are also available for you as you need. Okay, so we got our last regularly scheduled lab, so now it's getting stuff done. Remember, everything is due Friday. I'll say Friday at 4 is the deadline. So you're a no on any optional points, your food and urine project, and then Tuesday morning at 8 is the final. Okay, this is an antibody molecule. This is a particular type of antibody molecule belonging to a class of molecules called the G class of antibodies. We'll talk in a minute about the other classes of antibodies and what they are and how they differ. But this is kind of our model of antibody structure. What's this look a lot like? It should look a lot like the B-cell receptor. So, how is this antibody going to be produced? Well, the first thing that's going to happen is that a educated B lymphocyte, which has a receptor sitting out on its surface, that it's built by that gene shuffling process, is sitting in a lymph node and will have encountered its partner antigen, the one that matches the binding sites here and here on the antibody molecule or receptor molecule. Okay? Typically, we're going to see that for a B lymphocyte, that antigen will usually be presented to the B lymphocyte by an antigen presenting cell. And more often than not, that lymphocyte also will be stimulated by a cytokine that's produced by a T helper cell. So we're going to talk probably next Monday about that kind of complex interaction. But once it's received those signals, usually two, antigen binding and cytokine stimulation, then those B lymphocytes are going to start to produce antibodies. And as it turns out, this isn't the first class of antibody that they're going to produce, but this is the one that's going to be produced probably most abundantly as the process proceeds. We'll talk about that when we talk about classes of antibody. So, functional parts of the molecule. This particular region up right here, which consists of portions of the light chain and the heavy chain, is called the FAB region of the antibody. That is the antibody binding site for antigen. This region right down here, which is composed of constant regions of the heavy chain only, is designated as the FC region. FC means, in this case, that it is able to fix complement. So those are the two functional parts of the antibody molecule, the FAB and the FC. So the FAB region gives you your antigen specificity. That's what determines whether a particular antigen can bind to the antibody molecule or not. There's got to be a complementarity. And that complementarity is very much like an enzyme-substrate complementarity. There's got to be a fit. If there's not a proper fit, binding won't occur. The FC region will determine what we call the effector function of that class of antibody. An effector function is kind of a generalized function that all members of that class are able to do. So if there are a million different types of IgG antibody, each one of those will have specificity for a particular antigen. But all million of those will have this generalized ability to do something within the immune response. What are some examples of effector functions? Fixing complement. attaching to complement. Okay? Some can, some can't. Some classes can, some can't. Crossing the placenta. So you can get some maternal immunity to that developing fetus. Some antibodies are able to be secreted. When an immune cell is first activated, the first class of antibody that it produces is IgM. And we'll show you what the classes are in just a second. There are actually 5 classes of antibody. There's IgM, IgG, A, E, and D. So this is the first class of antibody that's being produced. Once a cell starts producing IgM, if it gets the proper cytokine signal, then it can sometimes switch into doing one of these other types of antibodies. This particular phenomena is called class switching. The most common class switch is from M to G. But other cells have the ability to go, depending upon the cytokines they're stimulated by, maybe switch from class M to class A, or class M to class E. Okay, so it depends upon the signal that you're getting, what switch you can do. We talked about memory cells, and we said that during an immune response, that usually, but not always, but usually, when a cell is stimulated, it can start producing plasma cells and memory cells. If you're a plasma cell, what are you going to do? You're going to start secreting IgM. If you get the proper cytokine signal, then that cell will then switch to forming class G antibodies, and now, by definition, it becomes a memory cell. So memory cells are those cells that can now produce class G antibodies. If you don't make the class switch, eventually, those cells will self-destruct and die by apoptosis. But if you have made this class switch because you've been exposed to the proper cytokines, then these memory cells will be very long-lived. Sometimes they'll last your lifetime. So this is the basis of memory, and memory is the basis of immunization. So this shows you what class switching involves. Okay, so here's our DNA, and this DNA has already been rearranged by gene shuffling so that a particular E region, a particular J region, a particular D region have been put together and associated with a particular C region. In this particular case, it's going to be the C region that codes for class M antibody. And those are going to be transcribed, and we're going to produce those particular antibodies of class M. But under the influence of a cytokine, probably produced by a T helper cell, but maybe produced by some other cell, the DNA can further rearrange. And this stretch of DNA, for example, can loop out these particular segments of DNA and cut them out. And then therefore join these segments to a C region that codes for class G antibodies. And now it's going to start producing class G. or if it loops out another segment of DNA, then it's going to associate the A region of the C gene, so we can now make class A antibodies. So it takes a further genetic rearrangement in order to switch classes and produce the new kind of antibody. And that class switching is really, really important. Here are the major classes of antibodies. Quite a bit of information in this particular slide. All of this I don't expect you to memorize, but there are some general kind of themes that I want you to be aware of as you look through this. Okay? One of the things you'll notice right here is that molecules have different sizes. And particularly, the class of antibody that's the biggest one is IgM. Look at its molecular weight, 900,000 compared to a much smaller IgG antibody. Well, why is that? Well, actually, class M antibodies are a pentamer. Okay? Penta means 5. Basically, it's 5 of these IgGs stuck together, and there is a polypeptide chain that puts those together to form that functional complex. So how many molecules of antigen can this bind, IgG? It's got a valence of 2, right? It can bind 1 here and 1 here. What's the valence of an IgM antibody? 10. Okay? It can bind simultaneously 10 molecules. IgG is going to be the antibody that's probably most important in especially long-term immunity. Let's take a brief look at some of its kind of characteristics. This is the one that has the longest life, and this is an average life in the serum. Look at here, 23 days, 6 days, 5 days, 3 days, 2.5 days. So these are longer lasting antibodies. Now here we're talking about the antibody, okay? Additionally, the cells that produce these, the memory cells, are very long-lived as well. So this is what gives you that sometimes lifetime immunity. Sometimes you need a booster shot, though, to kind of repopulate your cells, memory cell population, to kind of show them the antigen again. So for your diphtheria, tetanus, pertussis, about every 10 years you need to get a booster immunization to kind of re-up your memory cell population against that tetanus toxin and the diphtheria toxin and the pertussis toxin. Something like smallpox gives you pretty much lifetime immunity. Effector functions. IgG can cross the placenta, and so this gives us maternal protection for the fetus. So circulating maternal antibodies can cross the placenta and make sure that the fetus then has temporary protection while it's in utero. It will fix complement. This FC region of the IgG antibody can bind to the complement proteins in the blood serum if there's a pathogen present. What happens is the FC region binds to the pathogen. That then produces a complex that can fix complement proteins and ultimately result in that cascade reaction forming the attack complex. Not all of these antibodies can fix the complement. This particular type of antibody has a real affinity for phagocytic cells. So if antibodies have stuck to a pathogen, then the FC region of that can bind to the phagocytic cell. That brings the pathogen and the phagocytic cell together. Remember what we called antibodies that attach to the surface of pathogens and make them more likely to be phagocytized? Kidney signs? Opsonins, yeah. Okay. So because this can fix complement, it's an opsonin. So what's its long-term biological function? Well, it gives you long-term immunity. It gives you some memory. And we'll see in a minute later that it can do things like neutralize toxins and various viruses. Contrast that with IgM. IgM is the first antibody that's produced. But most cells will quickly switch over and start producing IgG. That's why the serum concentration of this antibody is much lower than the serum concentration of IgG. It's got a relatively short lifetime. Why do you suppose it can't cross the placenta? It's really big. Size. This Wilfix complement, this does not have a particular affinity for any particular type of cell. Any particular type of host cell. So whereas the IgG antibody has a real affinity for phagocytic cells, this, no real particular affinity for any host cell. So this is really a first response to antigen, basically. IgA is kind of interesting. IgA can occur in two forms. The bottom form down here, which is the monomer, is called the serum form of IgA. And so this is stuff that's produced by cells when they get the proper signal for class switching, that's then dumped into the serum. But the much more common form of IgA is the secretory form. This is a form of antibody that is secreted and particularly has an affinity for epithelial cells, and particularly for epithelial mucosa cells. So this antibody is constantly being pushed out into the surfaces of the lungs, for example, and the gut. And this provides first-line protection against bacteria that are commonly going to be found in those particular areas. Okay? When you inhale air, there's going to be bacteria. When you eat food, some bacteria will survive the stomach acid and proceed through your gut. And these IgA antibodies are sitting there on the surface of the epithelial tissue, ready to respond. This is a dimer of two monomer units, and there's a fairly complex polypeptide chain called the secretory component, which helps to allow this to transfer across the cell membrane and be secreted. This particular secretory piece is produced by whatever cell type is doing the secretion. So for example, IgA can be secreted into breast milk. Therefore, nursing mothers can give additional protection to the child while it's developing its immune system, because now the IgG protection that it was providing by placental exchange is no longer happening. So the mammary gland tissue actually produces this secretory component and hooks it on to these IgA antibodies that are circulating in the serum and makes this dimer, which is then secreted and gives you kind of surface protection on that epithelial tissue. It's a pretty neat system. IgD is kind of an interesting antibody. It's not really an antibody per se. This really is basically the B cell receptor, but they put it into this particular class of antibodies for some reason. I'm not sure why. But basically, functionally, this is the B cell receptor, and it looks a lot like the IgG if you see it. IgE, unfortunately, we're not going to get a chance to talk about much. And if we were a little bit further ahead, then I would love to have time to talk about allergies. IgE is the antibody associated with many allergic reactions. There are two types of allergic reactions. They're called immediate allergy reactions and delayed. Or immediate hypersensitivity and delayed hypersensitivity. Immediate hypersensitive reactions occur usually within 24 hours, sometimes almost instantaneously. Delayed hypersensitive reactions are slower, 24 to 48 hours. These particular delayed hypersensitive reactions give you some blistering type of reactions. You get bumps and things like that. This is a T cell response. And the reason you're getting these blisters and bumps is that these T cells are inappropriately attacking your own tissues. Anybody had a TB test? Okay. When you get a TB test, they scratch you with some of that allergen, and what they're checking to see is, do you have any prior exposure to TB? How do you know that? Because if you have, you will have some memory T cells that have been activated by prior exposure to TB. And those T cytotoxic cells will start affecting your skin tissue and causing that bumpiness that is a positive tuberculin reaction. Immediate hypersensitivity, which is most of the allergies that you're aware of, is a B cell response. And particularly, it is an IgE response. People who suffer from allergies, for some reason, overproduce IgE. The main role that IgE usually plays is it gives you protection against some multi-parasitic infections like various worms, for example. That's its major role in the immune system. But for some reason, individuals who have allergies are producing a whole bunch of this stuff in response to other stimuli, like pollen or insect venom. You can never become allergic to something on its very first exposure, because you need at least one exposure to stimulate B cells to start producing IgE. That particular phenomenon of stimulating B cells to produce IgE is called sensitization. You have to become sensitized first to the allergen. Maybe one exposure is enough to sensitize some people. Maybe it takes 10 exposures, or maybe no amount of exposures will sensitize another individual. But once you get a critical amount of IgE being produced, and once you become fully sensitized, then the next time that you see that allergen, IgE grabs onto it. And we've now got this antibody-antigen complex. And the IgE antibody complex has an affinity for mast cells and basophils. And when they attach to mast cell and basophils, they cause them to degranulate. And when they degranulate, they start releasing all these chemicals, including histamines. And what do histamines do? They trigger inflammation. So really, most of your allergies are actually inappropriate inflammatory responses induced by allergens that your body just shouldn't be reacting to. Okay? Plant pollen, for example, is not a threat. But for some individuals, plant pollen stimulates an overproduction of IgE, and once you get a critical amount of IgE circulating around, the next time you're exposed to the plant pollen, once you're sensitized, that complex then starts attaching to your mast cells and your basophils, causing degranulation, and you get your symptoms of your allergy, your inflammatory responses. So then you have to start saying, well, how can I counteract that? Well, you can take chemicals that counteract the effect of the histamines. You can take antihistamines. There are other therapies that have been developed to try to interfere with different parts of this allergic response. So class E antibodies basically, especially in the Western world, where multicellular parasitic infections aren't very common, have kind of developed into this new role of causing this bad response, the allergic response. So what's the importance of producing antibodies? Why do we bother producing them? Well, this particular slide gives you some of the reasons why secreting these antibodies can be beneficial in terms of your overall immune response. Upper left-hand corner. Here's a bacterial cell that has been tagged with antibody. Okay, if this is IgG antibody, then this region right down here, the FC region, is going to have an affinity for phagocytes. And it will attach to phagocytic cells, which will enhance the chance that this particular cell will be phagocytized. So this would be an example of opsonization. And it shows over here that they're now engulfed more easily. Another function of antibodies that is important is that antibodies can bind to viral receptors and block them. And if a viral receptor is blocked because it's already joined to an antibody, now it can't bind to a cell surface. And if a virus can't bind to a cell surface, it can infect it. So this particular phenomenon is called neutralization. It neutralizes the virus and prevents it from attaching to a host cell. This particular phenomena is called agglutination. And here what happens is one antibody can bind to more than one cell. Why? Because it's got at least 2 binding sites on it. If it's IgM, it's got 10. And this basically clumps together bacterial cells. The reason that that's advantageous is that it's more efficient for a phagocytic cell to grab a big clump than it is to basically get individual bacteria. So this agglutination really helps to facilitate more efficient phagocytosis. This is the basis of the blood typing reaction. Okay? In blood typing, what do? You take some blood, which contains your blood cells that have certain antigens on the surface, and you mix that with certain type of antibody. And if you get a clumping reaction or agglutination reaction, you can actually see it on the slide. And that tells you that your cells, if you mix it with A antibody and it clumped, that they have A antigen. Or if you mix it with B antibody and they clump, they have B antigen. Or if you mix it with both A and B and you get nothing, you have no antigens. And that gives you your blood types of A, AB, O, etc. Oh, one thing about it, I'm going to pass this around. If you were at the guest speaker, what's your name on there? We've also mentioned complement fixation. If a cell becomes attached to antibodies, then that antibody complex can fix complement and initiate a cascade, which causes cell lysis. Precipitation is very much like agglutination. Agglutination involves clumping together of whole cells. Precipitation involves clumping together of smaller soluble antigens. So a toxin, for example, might be precipitated by joining together with various antibodies. And again, once you form a complex of antigen and antibody, it's going to be more efficiently phagocytized. Okay, so what happens when you're exposed to an antigen? Well, this kind of complicated looking graph shows you the time course of what's happening in terms of total antibody production, IgM and IgG production, and shows you a little bit about what's happening to the various cells that are contributing to the formation of this antibody. The first time you're exposed to an antigen, you're said to suffer a primary response. Any subsequent exposure to that antigen would be a secondary response, and typically that results in an anamnestic response or a memory response. So what's happening? Well, first exposure to antigen, there's a latent period. It takes some time for things to get kicked up. Why? Well, think about the sequence of events. Let's say you're exposed to the antigen in your skin. So what has to happen? Well, some kind of an antigen-presenting cell is going to have to find that antigen and engulf it. And then it's going to have to travel from the skin through the lymphatic system to one of the secondary lymphoid tissues like a lymph node. And what's sitting in those lymph nodes? Educated B and T lymphocytes that have been produced in the bone marrow or the thymus, respectively. And once they've developed a receptor, they're now sitting in the lymphoid tissue waiting. So these antigen-presenting cells have to migrate to the lymph node and start showing that antigen around. Anybody respond to this? Anybody respond to this? Anybody respond to this? And when the final partner is found that matches up, and especially if we get other cytokine signaling, then that B cell can start cranking out antibody. First class M, and then when it gets some more cytokine signaling, or class G. So that's going to take time. Okay? And the only way you can get a class G produced is if you get a class switching event. So, what's your first response? A latent period, and then you get some IgM produced first, and then as class switching occurs and we produce memory cells, as those memory cells respond to the antigen, they can start producing IgG. And then eventually everything shuts down. So our total antibody production is a little meager, and there's been this latent period associated with developing that process. Now, maybe you'll produce enough antibody that you can survive that particular infection. And if you do, good, because then you have a memory of that. You have some of these persistent memory cells. But if your antibody production isn't high enough, or if the latent period is too long, and the pathogen is really virulent, you might die. Game over. If you've survived, you now have memory cells. The body will still be cranking out new educated immune cells and repopulating the lymph nodes constantly. So there'll be still some new cells that have never seen it before, but there'll be some cells that have seen it before. So what happens when you get a second exposure to the antigen? Well, the new cells have to go through the process of development of the antibody, start producing IgM. They eventually will class switch and produce more memory cells. But the memory cells that are already there can immediately start producing IgG antibody without that latent period. So IgG production is immediate almost because of the memory cells, and then all of a sudden these cells that have never seen the antigen before Well, they'll start producing M, and then eventually they'll class switch and add to this G, giving us a greater total antibody production. So this is the benefit of having seen the antigen before, is a faster response, probably within a day. It might take several days over here in the first response, this latent period, and you get a much higher total antibody production. So again, that's why we immunize. We want to show you the antigen in an inactive form the first time, so it doesn't matter if there's a latent period. The goal is to get the body to the point where it's producing memory cells, so that if you then see the antigen for real, then you've got a fast, rapid, robust memory response. We can break immunity down into categories as well. And there are two basic categories of immunity. This immunity that we develop in our lifetime is called acquired immunity. This is line 3. There is natural and there is artificial. Natural is developed as a result of experiencing the pathogen yourself, seeing the real thing in nature. Artificial immunity is produced through medical practices, i.e. immunization. Each type has two subcategories. There is active and there is passive. So if we look at natural immunity, the active form is the consequence of being infected. And then if you survive, you'll have a memory response. The passive form is the result of getting antibodies from another individual. In this particular case, the mother. In utero, it would be IgG antibody. Following birth, it would be, if you're breastfeeding, IgA antibody. This kid is developing his own antibodies, and that's why he's being protected. This kid is getting preformed antibodies from another individual. That's why it's passive. For artificial immunity, the active form is immunization or vaccination. And as a result of that intentional exposure, you stimulate the immune system. But because it's an inactivated antigen, you don't get sick. Passive artificial involves getting antibodies from another individual, not from breastfeeding, but from IV. Those particular antibodies are typically referred to as pooled gamma globulins. Gamma globulins are the class to which all antibodies belong. There are various types of globular proteins floating around in your blood, and the gamma class is the one that antibodies belong to. So where do you get pooled gamma globulins? Well, people donate blood or donate serum. And because of their past immunological history, one individual will have some active antibodies. Another individual will have different active antibodies. And they pool that serum, hoping that they get a good representation of possible antibodies that they can give to someone that is suffering disease. Why do they do that? Because that gives you pretty much immediate protection as soon as you receive the gamma globulin infusion. Because the antibodies are already there now, and they're working. And it gives you time to then weather out the infection and start producing your own active immunity. One of the instances where this is done not uncommonly is if there's a hepatitis outbreak. Because hepatitis is pretty nasty. And if epidemiologists trace a hepatitis outbreak back to, let's say, an infected food worker in a restaurant, then they can try to track down the people and say, you've been potentially exposed to hepatitis. And it's so serious that we would recommend you go into your doctor and get a gamma globulin shot. You're getting some antibodies that can fight that infection while your own system gets a chance to gear up. So, we can now start putting this together, and I'm just going to give you just a little preview of Monday where we kind of all put it together. So in adaptive immunity, there are really two overlapping, what we call arms of immunity. And that is the cell-mediated immunity and the humoral immunity. Cell-mediated immunity is T-cell mediated. Okay? So what's this going to do? It's going to kill pathogens and infected cancerous cells. Now remember, T cells are MHC restricted. MHC restriction ensures that they will only interact with cells, and in this particular case, bad cells, and result in their destruction. Humoral immunity is the other branch of the immune system, and this is involved in antibody secretion. The B cells are the ones that are actually producing antibody. But because most stimulating antigens are T-dependent, more often than not, a B cell needs some help to produce that antibody. It needs a cytokine. And that's produced by a T cell, a T helper cell. So up here in cell-mediated, it's really T killer cells that do the killing. T helper cells kind of slop over and are involved in humoral immunity as well. So that's why these two arms or branches of the immune system aren't separate at all. And the T cells, particularly the T helper cells, kind of join the two branches together. This is why HIV is such a devastating disease. HIV are specific for T helper cells. They attach to receptors that are on T helper cells, and they cause death of those cells. And if you destroy the T helper cells, you destroy not only the cell-mediated immunity, but you destroy the humoral immunity branch as well. So that's really important. So what we're going to do is we're going to look at cell-mediated immunity first and see, number one, how do we get these T killer cells activated? And as it turns out, they need a helper cell too. Once they're activated, how do they go about killing the infected cell? Talk about that. Then we'll talk about humoral immunity, and we'll say, okay, how do we activate B cells? Well, typically B cells need a T helper cell involved as well. But it's a different T helper than the ones that help these T killer cells. So we're going to differentiate between T helper 1 and T helper 2 cells. So there's going to be some cellular interactions you're going to have to keep straight. And there will be a few cytokines that you should be familiar with, because this chemical signaling is usually a very important part of that antibody production. That's what we'll do on Monday. Tuesday, we've got 2 days to talk about clinical applications of immunology, and that will basically be some information about various antibody-based tests that we can use to diagnose, and in some cases, treat diseases as well. So that's kind of where we're going to wrap up. So on your materials on Dropbox, this exam will be part of line 2, which will include inflammation, complement system, etc. Line 3, which we're talking about now, and then there's another set of notes and PowerPoint slides. It's fairly short, called serology, or applications of clinical microbiology. So that's going to be the exam 4. Okay? It's not comprehensive. There's plenty of information here for one exam. Okay, so lab exam will go over tomorrow, and if I have time, what I will do is I will, on the lab exam grade sheet, update your grade so you know what it is as of right now. It won't include any optional points because I only have those on to the end. Really, the only points standing out after you get the lab exam back will be your food and urine project, that's 35 points. Your exam is 100, and your unknown is 150. Okay. Pardon me? No, you're going to send the PowerPoint to me, and that's going to be the, you're not doing the presentation, no.